Welcome to the Concierge Care Health System podcast. The information shared in this podcast is for general informational purposes only and does not constitute medical advice. Listeners should be aware the content is not a substitute for professional medical advice, diagnosis, or treatment. Always consult with a physician or other qualified healthcare provider for medical questions.
Welcome to the Deep Dive. Today we're tackling a really significant topic. Major depressive disorder, or MDD. I mean, the scale is huge. Globally, we're talking about 280 million people affected.
Yeah, and just in the U.S. alone, something like 21 million adults had a major depressive episode just in the past year. It's pervasive.
But here's the thing that makes it so tough. The real challenge is the treatment. It's often this long, difficult process of, well, trial and error.
Exactly. Finding the right medication can take months, sometimes years. You try one, it doesn't work, or the side effects are too much, then you try another.
It's incredibly taxing. And the numbers bear this out, right? Something like 60% of patients, a huge number, don't respond to the first antidepressant they try.
That's right. And only about a third actually achieve full remission. That leaves this enormous treatment gap, a lot of people not getting the relief they need.
So you have this big variation in how people respond to drugs. And we also know there's pretty strong evidence that depression runs in families.
Precisely. And those two things together, the variability and the family link, that's really the core thinking behind looking at genetic factors.
The hope being that genetics could somehow guide treatment.
Exactly. Moving away from this, one-size-fits-all approach towards something more personalized, precision medicine, essentially. Can we predict who might respond better to which drug, maybe even at what dose?
So what does this actually mean for you listening right now? Well, today we're diving deep into genetic testing for depression. We want to pull out the key insights.
We'll look at the science, how these tests are actually being used or proposed for use in clinics.
And crucially, what the experts, the regulators, what are they saying about whether these tests are truly useful right now? And the ethics, there are big ethical questions here too.
Absolutely. So the goal is to understand where we really are today and maybe peek into what the future might hold in this area.
Okay, let's start with the scale again, just really ground this. MDD, it's not just, feeling down for a bit. The official definition from the DSM-5.
Right. It defines it as persistent, severe. We're talking symptoms like a constantly low mood, losing interest or pleasure in things that's called anhedonia.
Changes in appetite, sleep problems, fatigue, feeling worthless, trouble concentrating.
Even recurrent thoughts of death or suicide. And for a diagnosis, you need at least 5 of those symptoms for over 2 weeks, and one of them has to be that depressed mood or the anhedonia.
The impact is just, well, it's staggering.
It really is. The World Health Organization estimates about 5% of adults worldwide have depression right now. Lifetime risk could be up to 20% in some groups, average maybe 12%.
It affects women more, right?
Yeah, about 50% more likely than men. All of this makes MDD a leading cause of disability globally. The WHO even projects it'll be the single biggest cause of disease burden by 2030.
That sheer prevalence just highlights how urgent finding better treatments is.
Yeah.
And like we said, that trial and error medication approach, it's a major hurdle.
It prolongs suffering, increases risks, which is exactly why the idea of using genetics that promise feels so compelling.
Okay, so let's unpack the genetics then. You mentioned it runs in families. This isn't a new idea, is it?
Not at all. It's actually one of the most consistent findings in psychiatric research for decades. Twin studies, family studies, they all point to a significant genetic contribution.
Give us some numbers on that. How significant are we talking?
Well, for example, if you have a parent or a sibling with MDD, your own risk is about 2 to 3 times higher than someone in the general population.
So that translates to roughly...
A lifetime risk, maybe around 20–30% compared to the general population average. And twin studies, they're even more striking.
Identical versus fraternal twins.
Exactly. For identical twins who share basically 100% of their DNA, the concordance rate, meaning if one twin has it, the other does too, can be as high as 76%.
Wow, okay. And for fraternal twins?
Who share about 50% of their genes, like regular siblings, it's only around 19%. So looking at all the data, the overall heritability of MDD is estimated pretty robustly at somewhere between 40 and 50%.
40 to 50%. So nearly half the risk comes down to genes.
Mm-hmm.
That sounds... Well, it sounds quite deterministic. Does that mean your genes dictate whether you get depressed?
That's a really important question, and the answer is absolutely not. Genes are not destiny here.
How do we know that for sure?
Well, look back at those identical twins. Even with identical DNA, the concordance rate is high, yes, but it's well below 100%. One twin can develop depression, the other might not.
Right. So other factors must be playing a big role.
Exactly. Life experiences, stress, trauma, resilience, all those non-genetic factors are critical. It really supports this model we call the diathesis stress model.
Diathesis stress. Explain that a bit.
It basically means you might inherit a genetic vulnerability, the diathesis, but it usually takes some kind of environmental or psychological stressor to actually trigger a depressive episode. It's an interaction.
Okay, that makes sense. Now, how has the scientific thinking about which genes are involved actually changed over time? You hear about genes for depression.
Right. The early research, thinking back to the 90s, early 2000s, mostly used what's called the candidate gene approach.
Candidate gene, meaning they picked specific genes they thought might be involved.
Exactly. Based on what they knew about brain biology, like genes involved in neurotransmitter systems, serotonin, dopamine, etc.
Like that serotonin transporter gene, SLC6A4. I feel like I've heard of that one.
You probably have. It was studied intensely, SLC6A4, also called 5-HTT. It makes the protein that SSRI antidepressants target.
Ah, okay, so the logic was clear.
Very logical. And there was a hugely influential study back in 2003 by Caspi and colleagues. It suggested this gene interacted with environment. People with a certain version, the short allele, seemed more likely to get depressed after stressful life events.
Interesting. Any others?
Another big one was BDNF, brain-derived neurotrophic factor. That's involved in keeping neurons healthy and adaptable.
It all sounds quite plausible. So what happened? Did these hold up?
Well, here's where things got really interesting, and it marked a major shift in the field, methodologically speaking. Despite literally hundreds of studies on these candidate genes, especially SLC6A4, the initial findings, they just largely failed to replicate when bigger, more rigorous studies were done.
Failed to replicate? You mean they couldn't reproduce the results?
Pretty much. There was a landmark paper in 2019, huge study, hundreds of thousands of people. It found no statistical evidence linking that specific variation in the serotonin transporter gene, the 5-HTT-LPR polymorphism, to depression, not directly and not interacting with stress either.
Wow. So these celebrated findings just kind of evaporated. What's the lesson there for us?
It's a critical lesson about science, really, especially in complex areas like genetics. Promising early results often come from smaller studies, which can be prone to false positive.
Because of statistical chance.
Partly, yes. And maybe publication bias, positive results get published more easily. The biological ideas weren't necessarily wrong, but the effect sizes were probably tiny, if they existed at all for those specific single genes. As research standards got tougher, demanding massive sample sizes for validation, those early signals just faded.
So if it's not one or two main depression genes, what's the current understanding?
The consensus now is that MDD is highly polygenic.
Polygenic.
Yeah.
Meaning many genes.
Many, many genes, thousands probably. We know this largely from genome-wide association studies, or GWASS.
Right, those scan the whole genome.
Exactly. They look across the entire genome for tiny variations associated with the disease. And what they show is that the heritability, that 40–50%, comes from the combined cumulative effect of thousands of these genetic variants. Each one, on its own, adds only an infinitesimally small amount of risk.
So no single gene has a big effect.
Generally, no. The latest big GWS, looking at over 246,000 depression cases, found more than 100 distinct spots or loci on the genome linked to risk. But again, each contributes just a tiny bit.
Which brings us back to that idea of risk being probabilistic, not deterministic.
Precisely. It reinforces that diathesis stress model. Genes load the gun, perhaps, but environment often pulls the trigger.
Okay, so that's the underlying genetics. Complex, polygenic. How is this translated into actual tools that might be offered in a clinic? What are people actually using or trying to use?
The main application right now, the most developed commercially, is pharmacogenomic testing, PGX testing.
PGX. Pharmacogenomics, so using genes to guide medication choices.
That's the core idea, to try and personalize antidepressant selection, maybe dosing, and hopefully get around that slow trial and error process we talked about.
How do these tests actually work? What are they looking at in your genes?
They mainly look at genetic variations that affect how your body interacts with medications. You can broadly divide it into two areas.
Okay.
First is pharmacokinetics. That's basically what your body does to the drug, how it absorbs it, breaks it down, eliminates it.
Metabolism, basically.
Yes, metabolism is key here, especially by enzymes in the liver, like the cytochrome P450 system. You might see genes like CYP2D6 or CYP2C19 on reports. Variations in these genes can make someone a poor metabolizer, intermediate, extensive, which is normal, or even an ultra-rapid metabolizer.
And that affects drug levels in the body.
Exactly. It might suggest needing a lower dose or a higher dose, or maybe avoiding a certain drug altogether if you metabolize it way too fast or way too slow.
Okay, that's pharmacokinetics. What's the other category?
Pharmacodynamics, that's the flip side. What the drug does to your body. How it interacts with its target, like neurotransmitter receptors or transporters.
Does the genetic variation affect how well the drug binds or signals?
That's the theory. But frankly, the evidence linking specific gene variations in pharmacodynamic targets directly to antidepressant response is much, much weaker and less consistent than for the pharmacokinetic genes.
Got it. So practically, say I do one of these tests, usually a cheek swab, right? What kind of report comes back?
Right. It's usually a simple cheek swab or saliva sample, and there's a whole market of commercial tests now. Names like GeneSight, Genesip Assay, IDA Genetics might be familiar.
And they give you
What?
a list of drugs.
They typically use their own secret proprietary algorithms. They combine information from several gene variants, mostly the pharmacokinetic ones, maybe some pharmacodynamic ones, and they generate a simplified report, often color-coded.
Color-coded, like red, yellow, green.
Exactly like that. GeneSight, for instance, puts medications into a green category, use as directed. Yellow moderate gene-drug interaction, maybe adjust dose. Or red significant gene-drug interaction, maybe consider alternatives. The idea is to make it quick and easy for a busy doctor to look at.
Okay, that certainly sounds like it could be a game changer. Finally moving beyond trial and error. But I sense there's a but coming. What's the catch?
There usually is in science, isn't there? And yes, this is where it gets really interesting and honestly quite contested.
How so?
Well, the companies selling these tests, they often point to studies, frequently ones they sponsored themselves, that show some benefit. The GUIDED trial for GeneSight is a prominent example.
What did GUIDED find?
It found a modest but statistically significant improvement in depression symptoms and remission rates at 8 weeks for patients whose doctors got the GeneSight report compared to those getting standard treatment as usual.
Okay, modest but significant. That sounds promising, right?
It does sound promising on the surface. However, this narrative gets challenged when you look at larger trials, especially those funded independently, not by the companies.
The funding source matters. Tell me about those.
A key one is the PRIMECARE study. It was a large randomized control trial, nearly 2,000 patients in primary care settings. They compared giving doctors PGX test results versus usual care.
And what happened in PRIMECARE?
What's fascinating is, providing the test results did change the doctor's prescribing habits. They did prescribe more of the green light medications identified by the test.
Okay, so the test influenced behavior. That seems like a win.
You'd think so, but here's the crucial finding. That change in prescribing behavior did not lead to better clinical outcomes for the patients in the long run.
Really? No difference in how well patients did?
Not statistically significant, no. They looked at symptom remission at 24 weeks, which was their main outcome. No significant difference between the group where doctors got the test results and the group that didn't.
So the test changed what doctors did, but it didn't actually help patients get better more effectively. What does that imply?
That's the core of the debate right there. This gap between actionability, yes, the test gives you information you can act on, and clinical utility, does acting on it actually improve patient outcomes?
Why might that be?
Well, it suggests a few possibilities. Maybe those specific pharmacokinetic gene variations, while they definitely affect drug metabolism, maybe their overall impact on whether an antidepressant ultimately works for someone's depression is smaller than thought.
Smaller than all the other factors involved.
Possibly. Smaller than the complexity of the depression itself, other biological factors, psychological factors, environmental factors. Or it could mean that the proprietary algorithms these tests use to generate those red, yellow, green lights, maybe they aren't actually validated well enough to provide truly meaningful guidance. We don't know, because they're black boxes.
Okay, so that's pharmacogenomics for guiding treatment choice. What about the other angle, predicting the risk of getting depression in the first place?
Right. That's the realm of polygenic risk scores, or PRS.
Polygenic risk scores. How do they work?
The idea is to estimate someone's overall genetic predisposition to a disorder, like depression, by adding up the effects of many, many genetic variants across their entire genome. Remember those thousands of variants from the GWAS studies, each with a tiny effect?
Yes. So PRS sums up all those tiny risks.
Essentially, yes.
Yeah.
It takes the results from large GWAS studies and calculates a score for an individual based on which risk variants they carry, weighted by how strongly each variant is associated with the disease.
Is that approach more promising for clinical use?
For research, PRS are definitely a valuable tool. They can statistically tell the difference between large groups of people with depression and large groups without it. They help us understand the genetic architecture.
But for predicting risk in one individual patient sitting in front of you?
That's where the problem lies. The transition to using PRS for individual patient care for prediction or diagnosis hasn't happened. The main issue is their predictive power at the individual level is still quite low. Meaning, even if you have high PRS for depression, it tells you your risk is elevated compared to the average person, but it absolutely cannot predict if or when you personally will develop depression. The amount of risk explained by current PRS is just too small for that kind of individual prediction.
And are there other issues with PRS?
A major one, yes. Equity and generalizability. The huge GWS studies used to develop these PRS have overwhelmingly relied on data from people of European ancestry.
So the scores don't work as well for people from other backgrounds?
Exactly. Their accuracy and usefulness drop off significantly when applied to individuals from non-European ancestral groups. This is a massive issue, raising serious concerns about health equity. So for now, using PRS for MDD in a clinical setting is definitely not recommended.
Okay, so we have these PGX tests with contested utility and PRS not ready for prime time. Given all this, what are the official bodies, the regulators, and professional groups actually saying?
This is really telling. Despite the commercial marketing, there's actually a strong consensus among major regulatory and professional organizations. And it's a consensus of caution, which contrasts sharply with some of the claims made by the test manufacturers.
Let's start with the big one, the FDA in the U.S. What's their position?
The Food and Drug Administration has been pretty clear and firm. Back in November 2018, they issued a safety communication.
And what did it say?
It explicitly warned the public and professionals that, quote, the relationship between DNA variations and the effectiveness of antidepressant medications has never been established.
Never established. That's strong.
It is. They stress that making treatment decisions based on unproven claims from these tests could lead to patients getting inappropriate treatment, stopping needed medication, and facing, quote, potentially serious health consequences.
Did they take any action?
Yes. They even sent a warning letter to a specific genomics lab in 2019 for illegally marketing tests that claimed to predict patient responses to antidepressants without having gone through FDA review and approval.
Okay, so the FDA is very cautious. What about the main professional body for psychiatrists, the American Psychiatric Association, the APA?
The APA basically echoes the FDA's stance. Their Council on Research has done systematic reviews of the evidence. They concluded back in 2018, and they just updated this in a report in May 2024.
So very recent.
Very recently, and their conclusion remains the same. There is currently insufficient evidence to support the widespread use of PGX tools in clinical practice for depression treatment.
Insufficient evidence. Even with newer studies like GUIDED coming out, why are they still so skeptical? What are the main sticking points for them?
Now, that's a key question. Why aren't these newer studies convincing the experts? The APA points to several persistent methodological flaws in the research base.
Such as?
A major one is the lack of blinding in the trials.
Blinding? Meaning the doctors and patients know they're getting the genetic test.
Exactly. Almost none of the trials have been double-blind, where neither the patient nor the doctor knows if they received the real PGX test result or some kind of sham or control information.
Why is that so important?
Because without blinding, you absolutely cannot separate the true pharmacological effect of choosing a drug based on the test from the powerful effects of placebo, expectancy, and just getting more attention because you're in a trial. People expect the genetically guided treatment to work better, and that expectation itself can influence outcomes.
That makes total sense. What other issues?
Second, the control groups used in most trials are often inadequate. They usually compare PGX-guided care against just treatment as usual.
And treatment as usual isn't necessarily great.
Well, it's highly variable. It's not usually a standardized evidence-based treatment algorithm. A much stronger comparison would be to test PGX guidance against an already optimized, structured approach to prescribing, maybe one based on clinical guidelines.
See if the genetics adds anything on top of already good care.
Precisely. And the third major point is the pervasive industry funding. The vast majority of studies that report positive results for these PGX tests are funded, directly or indirectly, by the companies selling the tests.
Which inevitably raises concerns about potential bias in how the studies are designed, analyzed, or reported.
Absolutely. It doesn't automatically mean the results are wrong, but it requires extra scrutiny and independent replication, which has often been lacking or contradictory, like with PRIMECARE.
And you mentioned those black box algorithms earlier. That seems like another hurdle.
Yes, it's a fundamental problem for evidence-based medicine. These commercial tests use proprietary algorithms' trade secrets to combine the genetic data and spit out those recommendations.
So researchers can't check their work.
Exactly. Independent scientists can't scrutinize, validate, or replicate the specific logic the test uses. Clinicians are essentially being asked to trust a confidential formula. That lack of transparency is a major obstacle to scientific acceptance and trust.
Okay, so the scientific and regulatory hurdles are significant. But let's say, hypothetically, the science did become rock solid. There are still other layers here, right? Ethical, legal, social issues.
Oh, absolutely. Genetic testing in psychiatry, perhaps more than in other areas of medicine, opens up a whole Pandora's box of complex issues.
Where do we even start?
Well, informed consent is a big one. Getting truly informed consent for these tests is incredibly difficult.
Why?
Because genetic information, especially for complex traits like depression risk, is probabilistic. It's about slight shifts in odds, not definite predictions. It's complex information to convey and for patients to grasp fully.
So there's a risk of misinterpretation.
A huge risk. Patients might see a bad result and feel doomed, leading to genetic pessimism. It's in my genes, I can't escape it. Or they might get a good result from a PGX test and expect a magic bullet cure, setting them up for disappointment.
Which points to the need for good counseling.
Critically important. Comprehensive genetic counseling is needed to navigate this. But honestly, we just don't have enough trained genetic counselors to meet the potential demand, and many doctors haven't had the training to provide that level and detailed counseling themselves.
Okay, informed consent is tricky. What about stigma and discrimination? Mental illness already carries a heavy burden.
Right, and adding a genetic label on top could potentially make it worse. It might reinforce outdated, harmful ideas about mental illnesses being permanent. Inescapable biological flaws could lead to more shame, more hopelessness.
And discrimination, like with jobs or insurance.
That's a crucial question. Now, in the U.S., there is some protection. The Genetic Information Non-Discrimination Act, GINA, was passed in 2008.
Gina, what does it do?
It provides federal protection. It prohibits health insurers from using your genetic information to decide if they'll cover you or how much to charge you. And it stops employers from using genetic information in hiring, firing, or promotion decisions.
Okay, that sounds pretty good. So you're protected.
Mostly for health insurance and employment. But here's the really important catch, the significant gap in GINA's protection.
What doesn't it cover?
It does not extend to life insurance, disability insurance, or long-term care insurance.
Wait, life, disability, long-term care. Those seem pretty crucial, especially if you're concerned about a potentially chronic condition like MDD.
Exactly. For someone found to be at high genetic risk for a condition that could be disabling or affect the lifespan, those are precisely the types of insurance that become most vital. But insurers in those categories can potentially ask for or use genetic information to deny coverage or charge much higher premiums.
That creates a massive dilemma for someone considering, say, a predictive PRS test.
It's a high-stakes gamble. You might gain some health insights maybe, but you risk making yourself uninsurable for key aspects of financial security later in life. That's a very practical and significant ethical barrier for you, the listener, to really think about.
Wow. Okay. What else? Privacy must be a concern too.
Huge concern. Genetic information is unique because it's not just about you, it's inherently familial. Your genetic results have implications for your parents, your siblings, your children who share parts of your DNA.
Creating potential conflicts.
Definitely. Your right to know your genetic status might clash with a relative's right not to know their own potential risks if your results imply something about them. And beyond the family level, just the fact that this deeply personal data is being collected, stored digitally, often by commercial companies, it raises profound questions about data security, breaches, potential misuse, or even re-identifying people from supposedly anonymized data.
So wrapping this all together then, what's the big picture here? Where do we stand?
Well, undoubtedly, genetic research has fundamentally changed how we understand major depressive disorder. We now clearly see it as a complex brain disorder with a real heritable biological basis, specifically a polygenic architecture.
Which does open the door to a new way of thinking in biological psychiatry.
Absolutely. It holds out this tantalizing promise of eventually moving towards treatments that are precisely tailored to an individual's underlying biology. That's the dream of personalized medicine.
But how do we get closer to that dream? If current tests aren't quite there, what's next?
I think the consensus emerging now, looking at the bigger picture, is that the future probably lies beyond just looking at genomics, the DNA sequence in isolation.
Meaning we need to look at more than just the genes themselves.
Exactly. The future likely involves what's called a multi-omics or systems biology approach. It means integrating information from many different biological layers.
Like what kind of layer?
So genomics, yes, your basic DNA blueprint, but also epigenomics, the chemical tags on DNA that control which genes get turned on or off. Transcriptomics, which genes are actually being actively expressed into RNA. Proteomics, the proteins being produced. Metabolomics, the small molecules involved in your metabolism. Even microbiomics, the community of bacteria in your gut, which we now know influences the brain.
Wow, that's a lot of data. Combining all of that.
Yes, using sophisticated tools like AI and machine learning to integrate these huge data sets. The hope is to build much more accurate, much more nuanced models of who is at risk, how to diagnose different subtypes of depression, and crucially, who will respond to which specific treatment.
So instead of just depression, you might identify subtypes based on a whole biological signature.
Imagine that. Identifying, say, an inflammatory subtype of depression based on its specific genetic profile, combined with certain epigenetic marks, plus maybe specific protein and metabolic signatures in the blood. That kind of specific biological profile would be a much more promising target for a tailored intervention than the very broad MDD diagnosis we use today.
That potential sounds enormous, but you're saying we're clearly not there yet for clinical practice right now.
Definitely not there yet. It's exciting research, but it's still research.
So what's the crucial takeaway for our listener today, if they're thinking about these tests or hear about them?
I think the main message has to be that for any genetic tool, whether it's for guiding treatment or predicting risk, to become a standard part of clinical care, the bar for evidence needs to be set much, much higher than it is now.
Higher standards for the research.
Absolutely. We need large, well-designed trials. They have to be fully blinded, as we discussed. They need appropriate control groups comparing against best current practices, not just treatment as usual. They need to be funded independently to avoid bias.
And crucially, they need to include diverse populations.
Yes. Critically important. The research must be done in diverse, representative populations to make sure the findings are equitable and actually apply to everyone, not just one ancestral group. So for this present moment, when it comes to using these genetic tests widely for depression in the clinic, the verdict really has to be one of caution, patience, and an unwavering demand from patients, clinicians, and policymakers for better, more rigorous evidence.
The potential to help people is real, but we need to get it right.
Exactly. The potential to alleviate a huge amount of suffering is there, but we can only realize that potential if we prioritize genuine patient welfare and solid scientific evidence over, premature commercial hype or wishful thinking.
So looking ahead, perhaps the most important question for all of us is, how do we make sure this incredibly powerful genetic technology is developed and ultimately used in a way that's responsible, ethical, and truly benefits everyone equitably?
Concierge Care, providing world-class, personalized care for patients and members. Taking a proactive approach by offering its free digital health clinic for anyone in need of these services. Reach out to us via our website, www.health.conciergecare.net, or give us a phone call at 786-809-2345.